Welcome back, everyone, from lunch. Hope you had a very nutritious experience at lunch. And so one of the jokes I commonly use when we sort of open the summit is, don't we all look great for 62 years of being at this here at our 62nd annual summit? But I have the deep honor today to be able to honor two people who have truly been at this for 62 years. This is our induction ceremony into the Nutrition Hall of Fame. And so you're in for a real treat. We're honoring today two nutrition scientists who have done tremendous work in the world and on whose shoulders all of us stand and who've paved the way for us here today. So it's a real treat and an honor for us to be able to be here and to celebrate these two nutrition scientists. The award for the Nutrition Hall of Fame is bestowed upon individuals who have demonstrated monumental achievement in the field of nutrition, advancing the science of nutrition, and whom are recognized by their colleagues as trailblazers in the field. Dr. George Brewer and Dr. Ananda Prasad have changed the way we think about micronutrients, particularly copper and zinc. First, Dr. George Brewer, MD, MACN. Dr. Brewer received his medical degree from the University of Chicago and is the Sellner Emeritus Professor of Human Genetics and Emeritus Professor of Internal Medicine at the University of Michigan Medical School. He is currently co-director of VetGen Laboratories and co-director of Cypress LLC. He served as our president of this organization when it was called American College of Nutrition from 1996 to 1997. He received the ACN Award for Distinction in Outstanding Research in Nutrition in 1989 and the ACN Mastership Distinction for the very highest level of scientific achievement in the organization in 1999. Other awards include a USPHS Career Development Award, Distinguished Alumni Award from the University of Chicago, a Certificate of Appreciation from the Wilson's Disease Association, a Rowland Award from the International Society for Trace Element Research in Humans, an Honorary Doctor of Science from Purdue University, and he is Fellow of the Morris Animal Foundation and the American Association for the Advancement of Science. He's done seminal work in our understanding of copper metabolism and its role in human health. Second, Dr. Ananda Prasad, MD, PhD, MACN. Dr. Prasad, also a past president of this organization, has been a professor at Wayne State University since 1963 when he took a position as director of the Division of Hematology, a post he held until 1984 when he became the director of the Division of Research. Dr. Prasad has also been a professor of medicine at Wayne State from 1968 until the present. He was appointed as Distinguished Professor of Medicine, Division of Hematology Oncology in 2000. Dr. Prasad's pioneering studies from the Middle East in the early 1960s established for the first time the essentiality of zinc for human nutrition and showed that its deficiency occurred in humans. The impact of this discovery has been immense, including the establishment of an RDA for zinc in 1974 and mandatory inclusion of zinc in TPN fluids in 1978. His work has truly saved countless lives. Its use in children with acute diarrhea has decreased mortality substantially, and its use in the elderly with age-related macular degeneration for the prevention of blindness. Dr. Prasad is author of 12 books and over 300 scientific articles. Dr. Prasad's papers have been cited as classics in the field. One paper was republished as a nutrition classic and another as a landmark article. In 2011, he received a prestigious Mahidol Award from Thailand for his contributions in the field of zinc metabolism. And he received an award from the American College of Physicians for outstanding research in medicine in 2007. And it is one of the true personal and professional honors of my life to be able to induct Dr. Brewer and Dr. Prasad into the Nutrition Hall of Fame today. Not only are they incredible giants in the field of science, they're incredible human beings. They have been amazing leaders of this organization for decades, but also at a crucial juncture of the organization 10 years ago. They joined our leadership team in really positioning the organization to now become and blossom and flower into all that is here today. So there's a deep personal respect and gratitude that I have for both Dr. Brewer and Dr. Prasad. I'm honored not just to call them colleagues, I'm pleased to be able to call them my personal friends. Some very interesting, continuing amazing work that these gentlemen are doing. Just one example is Dr. Prasad in the very early stages of COVID was infected with COVID before there were any treatments or vaccines. And he, through his own therapeutic regimen, brought himself back to vibrant health at the ripe age of 92 with his nutritional and related interventions, one of which was, of course, zinc. So it's a beautiful thing to come full circle after having saved countless lives with his discovery for it very possibly to have saved his own life at the age of 92. And Dr. Prasad continues to do amazing cutting-edge work in the field to this day, now at the ripe age of 93, and is just an amazing human being and scientist. We're very pleased to be able to provide to you right now a video tribute to Dr. Prasad, and it features Dr. Brewer in it. It was produced by Dr. Prasad's grandson, John Eckenrode. So let's roll the video.
Dr. Prasad is really the founder of zinc deficiency.
Everyone in the world knows Dr. Prasad as a specialist in zinc.
Zinc deficiency probably involves about a third of the population in undeveloped countries, particularly in young children, and causes a lot of fatalities.
The fact that people in developing nations now are able to survive and thrive and grow and live normal lives is huge.
In my experience, he's the person who's achieved the most in terms of combining really good science with an impact on public health that I've ever known of. So he's really been a giant in the field.
I came of a very large family. I had 2 brothers, 4 sisters, and they were all very kind and affectionate to me. When I was 6 years old, I wanted to go to the same school where my older brother was. He was in this district school which I started from 4th grade to 12th. I was clearly not prepared. I did very poorly the first term. And my father told me that you have to come back home and get prepared. But I told him that I want to try one more term, and I worked very hard, and I stood first in my class. And after that, I always stood first. We were not familiar with the United States in India at all. But after independence, they were trying to get Indians familiar with what was going on here. The concept was developing in USA that physicians should not only be clinicians taking care of patients, giving medicine, but they should also be scientists. Because if there is a disease, if there's a problem, the physicians will then be able to take it in the laboratory and work as a scientist to understand. And I thought that it was a great program. After I finished my training with Cecil Watson, Professor Hobart Ryman, he was chief of medicine at Jefferson in Philadelphia, He was also a personal physician of Shah of Iran, and the Shah invited Robert Ryman to go to Shiraz Medical School and set up a medical curriculum just like the American medical schools. And he accepted that position. Now he wanted a young man to help him do this job in Shiraz. So he contacted Cecil Watson, and Cecil said that since I was waiting for a position in India, maybe I could go to Iran and help him. I accepted his challenge, went to Iran, and there I discovered the zinc deficiency problem.
The story with Dr. Prasad and the zinc is a fascinating story, and it's a really fun story.
He traveled to Egypt and Iran and with an American group, and they were puzzled by the villagers in these countries. A lot of the young men turned out to be dwarfs.
And one of the habits these young men had, as he described it, was they were eating clay, partly to overcome their hunger, but partly to overcome some of their mineral deficiencies.
What he realized was that by eating the clay, the dirt was actually leaching the zinc out of the children.
It was already known in the literature that zinc was essential for the growth of rats and plants and pigs. I thought that, well, if zinc is a growth factor for the animals and the plants, perhaps I'm looking at zinc deficiency in human.
It turned out that when he gave these dwarfs zinc, these young 25-year-old dwarfs grew from pre-adolescent boys to adult men.
All my colleagues got together, we celebrated that we have really discovered a very important element for human health.
Ananda is a family man. He's very excited and loves and talks a lot about his children and grandchildren.
Others know him as a scientist, but Ananda would first refer to himself as a husband and a family man, a father, and then as a scientist.
I have very close relationship with all my children, and my wife has been a great support for me in every respect.
I view him and his wife, Arabella, as good friends. They've traveled the world in spreading the word about zinc, and he has inspired the work of a lot of young scientists. I was one of them.
He reminds us all that we're all really students. It doesn't matter if you're assistant professor or professor at the beginning of your career or in the middle, we're all students.
It gives me great satisfaction and pleasure to look at that here I was many years ago. I opened up the whole world of zinc, completely unknown, but this legacy is going to last.
Unfortunately, Dr. Brewer is unable to be with us today, but we extend our gratitude and esteem to him, and we'll be sending him his Nutrition Hall of Fame award. But we have a treat in that Dr. Prasad is with us today, along with his wife, Arabella, and his family. I'm honored to welcome Dr. Prasad to the podium to induct him into the Nutrition Hall of Fame. And we're in for a treat in that he is going to give his Hall of Fame lecture titled Discovery of Zinc as an Essential Element for Humans, Impact of this Discovery on Human Health, and Biomarkers of Human Zinc Deficiency. So, I now bring to the stage to formally induct into the Nutrition Hall of Fame, Dr. Ananda Prasad.
you, Mike. Thanks a lot.
Okay, wonderful.
Okay,
Thank you very much.
thank you. Well, thank you very much for asking me to give this lecture. As you heard, that I was trained under Cecil Watson in Minnesota as a physician scientist, and that training really helped me to understand many diseases. I kept on not only looking at the clinical aspects, but also I looked at the biochemical, molecular, immunological, and that's what my life has been all about until now. I found excitement at every step. May I have the next slide? Can you change the slide for me, Sheila? Yeah. As you heard, I went to Shiraz, Iran, and at the medical center Grand Round, Dr. Nadimi, who was my resident, presented a patient who was 21 years of age, and he was very much growth retarded. He looked like 10-year-old boy. He had big liver and spleen. He had no facial hair, no beard, and he was also eating clay, something like a pound of clay every day. And this combination, the reason why the resident presented the case was because I was a hematologist and he was very iron deficient. And when I heard the case, I was completely dumbfounded. I've never seen any description like this anywhere in the literature or in the textbook of medicine. And so, first of all, I determined that he was iron deficient, to be sure. No question about it. Hypochromic microcytic anemia, hemoglobin was only 8 grams. But the real problem was that he had no blood loss. We had investigated very carefully. And if you read the textbook of medicine, it says that adult males do not become this much iron deficient without blood loss. So that was my first problem. How did he become iron deficient? But the second problem was that even if he was iron deficient, that could not account for growth retardation. For instance, if you take rats, mice, elephants, pigs, make them iron deficient, nothing happens to the growth. They become iron deficient. They have hypochromic macrocytic anemia, but no effect on growth. Well, clay eating was another problem, and I just looked into the literature for all the essential elements that we knew. There were 6 transitional elements that we knew were essential for human, and there was one element, zinc, I found very interesting, that it was essential for the growth of Aspergillus niger, a fungus. This was reported from Professor Leon at Lyon University in France. And he reported it in 1869, but it remained completely unnoticed. 40 years later, they found that indeed plants also become zinc deficient and they do not grow. And then the laboratories in Wisconsin, they produced experimental deficiency of zinc in rats, and they found that growth was affected very much in those rats. So here I was in 1958. I came to Shiraz, and I thought to myself that perhaps I'm looking at a patient who is zinc deficient. But the problem was that if you looked at the textbook of medicine, looked at all the literature, talked to all the nutritionists, they all told me that zinc is not essential for human, and you will never find a zinc deficient human subject. So I was facing a tough cliff to climb. But we did some studies in Iran itself. One thing we did was to put them in the hospital, gave them good diet, took them off clay eating, and the nutrition was corrected, and we gave them 15 milligram of zinc a day. And to my great surprise, we found that the growth retardation was completely corrected in this patient. Well, it turned out I asked my resident that, is this the only patient that you have in Shiraz, or there are more? Because the local physicians told me that it is, of course, hypopituitarism. But he said that, Dr. Prasad, I can bring as many patients as you want. And I told Professor Shea that, it could not be that in Shiraz you have an epidemic of hypopituitarism, that so many patients are growth retarded. And of course, this remark was not taken very well by the professor. He thought I insulted him, but I really, it came out of my mouth immediately, and disregarded the idea of hypopituitarism. And I started to focus on the fact that perhaps it's nutritional deficiency of zinc. And perhaps clay eating added to this problem, because clay also can bind a lot of metals and produce deficiency. We took care of several patients in Shiraz. We published our first paper in the American Journal of Medicine, 1961, suggesting that this was a zinc deficiency problem. And obviously, after publication, I got several comments. Many of them were, they thought that maybe I was correct, but many were very controversial, and they really outright rejected my idea. Well, one professor from Vanderbilt University, Dr. Darby, he took it very seriously, invited me to come to Vanderbilt University, and he said he will make facilities for me to study zinc deficiency in Namru 3, Naval Medical Research Unit in Egypt. And I will be able to study zinc metabolism very well and prove whether or not zinc deficiency occurs. I took that challenge, went to Egypt, and the measurement of zinc in plasma red cells was exceedingly difficult. The only way you could assess zinc was to extract it and work for it all day and use dithiazone technique to assay zinc. And literally, at that time, in the entire world, there was only one laboratory, and that was Burt Valley in Boston, who was able to study zinc metabolism. And a friend of mine, Professor Charlie Davidson, said that I will ask Burt to assay zinc in these patients and give you the results. As it turned out, that Burt never did the assay, and now I was ready to leave Iran and now Egypt coming. And so I decided that, well, if I have to do some study, I must do it myself, set it up. And that's what I told Darby to help me with. NIH director was there, and Galloway, who was Admiral of US Navy, was also there. They all met with me in Namru 3. And they said that we'll give you all money, all facilities, everything that you need to study zinc. And they asked me how much money I needed. I told them that, well, just give me 2 research assistants and I will work with them. And they said that, I did not come from Boston, from Bethesda, Maryland, to give you just $20,000 grant. And so Arnold Schaefer sat down with me, and pretty soon my budget went up to around $90,000. Big money during at that time, if you imagine what the NIH funds are. Well, I collected about 20 dwarfs there and put them in the metabolic unit, monitored every bit of what they were eating, and acid, all the things that we could, vitamins and minerals and so on. And I was able to report that indeed plasma zinc was decreased in these patients. And when we supplemented them, to my great surprise, I found that they gained 6 inches of height in the first year. Actually, first time Dr. Sandstead came to me and he said that, Ananda, this boy that I gave him zinc, he cannot fit into his cap, he cannot fit into his clothes, and he's very happy. He has grown beard and mustache, and it feels like he has gone through puberty. And I had a party at my home that evening. I knew that I have really hit a very important event in my life, showing that zinc is essential for human. Well, I came back to published several papers there. The most important paper that I published was in Journal of Lab and Clinical Medicine, and that became Nutrition Classic, and showing that indeed zinc deficiency occurs. And this paper was again republished 50 years later almost as a landmark article in Journal of Lab and Clinical Medicine. So I was then invited by Richard Wink to come to Shiraz from Egypt to Detroit, Michigan, as chief of hematology. I came in 1963, and my first effort was to find out how is it that zinc affects growth. There was no evidence that zinc was involved in any growth factor and so on and so forth. So we looked at the growth aspect. We looked at the growth hormone in pigs in collaboration with Michigan State University. And it turns out that the growth hormone was not affected at all by zinc deficiency. This was, later studies showed that it was the incorporation of thymidine in the DNA molecule that was affected adversely. And the enzyme required for that was deoxythymidine kinase. And this was absolutely new and astounding observation. And we later found in our studies that the gene expression of deoxythymidine kinase was zinc dependent. And if this did not happen, TTP was not formed and DNA was not formed and cells did not multiply and there was growth retardation. This was confirmed later by other investigators also. Next slide. And so we postulated that the role of zinc in growth was because it was affecting deoxythymidine kinase, and TTP was not being generated, and therefore DNA was not formed and cells did not divide. And so that's the story about the growth. The other thing that I was very concerned about is I found when I was in Egypt that these patients, growth retarded, zinc deficient subjects, they never lived beyond the age of 25. And I went to all the villages and all and asked questions as to why is it that they are not living. And I was told by the physicians that they die because of infection, pneumonia, meningitis, and things like that. And that just struck me that perhaps cell-mediated immunity was the major effect due to it was affected adversely because of zinc deficiency, and that's why they were getting infection. And that became my primary goal, to find out how is the immune deficiency affected due to zinc deficiency. Well, one way to study was to develop a human experimental model. It was, as you know, that not too many studies have been done in nutrition where you have developed experimental model, and the reviewers, they were critical of me that I wanted to restrict zinc in human diet when I'm saying it's essential. But to my great help, FDA, USDA, NIH, Kellogg Foundation, all these people, they said that, no, we want you to do this work. And we had no CRC unit at Wayne, so I went to University of Michigan, and that's where I got to meet Dr. Brewer, because he was in human genetics, and we had some collaboration in sickle cell disease and others, and I told him that I would like to study zinc deficiency. So we got 2-bed supported by NIH CRC unit to develop experimental model. And truly, the knowledge that I got by this model is phenomenal. I found out that the people all over the world, the only thing they can do is measure zinc in the plasma. And plasma was the least sensitive marker for zinc deficiency. We found that there was a marker, thymolin. This is a thymic hormone, and this thymolin, when it gets zinc in the thymolin, it becomes active, and it becomes active and develops T helper 1 cells and generates interleukin-2 messenger RNA. And this interleukin-2 messenger RNA activates natural killer cells, T cytotoxic lytic cells, and it had tremendous effect on cell-mediated immune function. Also, interferon gamma was affected adversely, and interleukin-2 messenger RNA if there is zinc deficiency. And later, our studies have shown that indeed the T memory cells is also dependent on zinc status. And we can, now this is the overall slide that I'm showing how things are working in the cell-mediated immunity. On the top left, you can see the thymolin, and thymolin must be activated by zinc, and then IL-2 messenger RNA is generated, and that generates interleukin-2 interferon gamma. But one very surprising event that we saw, and that I was completely astounded to see, that the TNF-alpha, interleukin-1 beta, IL-6, these are the inflammatory cytokines. They were going up in mild deficiency of zinc. These patients had no infection, no toxicity, no chemicals. They were just experimentally made zinc deficient by restricting zinc in the diet. And this stimulated monocytes macrophages to generate all the inflammatory cytokines. And that was a very remarkable observation. And I think that perhaps the subjects that we saw in Middle East and all, they had a zinc deficiency, and they were having all these inflammatory cytokines, and that's why they used to get all the infections, and they used to die by the time they were 25 years of age. Next slide. Now, this slide is, yeah. Well, this is the, okay. I want to tell you a little bit about here that if you have monocyte macrophages, next slide, maybe I'll show it better, ROS, yeah, yeah. If the cells are exposed to inflammation, for instance, bacterial inflammation or viral infection or chemical and all, they will activate monocyte macrophages to generate all kinds of inflammatory cytokines. In the next one, I'll show it. Next slide. Okay, well, okay, next slide. Yeah. Okay, well, yeah, the major effect would be due to zinc deficiency. You will have a decrease in T cytotoxic lytic cells and also decrease in natural killer cells and interleukin 2. And on the other hand, you have the inflammation that creates a lot of problems in human health. Okay, next one. Yeah. I'm sorry, I'm not able to read this one, but move on. Oh, yeah, okay, this is very, yeah, I want to just briefly mention here that when I first suggested that zinc deficiency might affect enzyme activity, all the biochemists said that you are completely wrong. You need so little zinc for all the enzyme function that nature will never give up that zinc with mild deficiency. But that's what I learned from my human model, that deficiency was very mild, just we restricted to 2 milligrams a day. And in spite of mild deficiency, several enzymes were affected. Most importantly, deoxythymidine kinase, which affects the growth and development. And then in 1989, Klug reported that the transcription factors, the one that dictate the generation of messenger RNA of several proteins, they were also a very important role of zinc. And I was at a meeting in Turkey, and I suggested to the scientists there that perhaps we should be looking at these transcription factors. And everybody laughed at me and they said that, no, Dr. Prasad, it will never happen. Transcription factors need very little zinc, and it's very essential for messenger RNAs, and you will never see any effect. But my work for the last 30, 40 years has been just to look at these transcription factors. We have already shown that IL-2 messenger RNA is affected. AP transcription factors like NF-kappa B, AP1, SP1, and in T helper 1 cells, and many transcription factors that are involved in generation of T helper 1 cells, including Tibet, and all these are affected very adversely, no question about it. And basically what is happening that if there is an infection, that molecule goes to the membrane, cell membrane, and it generates various effects on the cell and activates NF-kappa B in the monocytes macrophages. And these monocytes macrophages then really get affected, produce inflammation, inflammatory cells, and cause various health effects. Now, with respect to genetics, I want to tell you that it was reported that patients with acrodermatitis enteropathica, they are very zinc deficient, and they die of infection by the time they were 20, 30 years of age. But once we realized that it was zinc deficiency and they were treated promptly and adequately, then they lived. Actually, it turns out that this disease is caused by a transporter ZIP4, which brings the zinc inside the cell from outside. And ZIP4 mutation causes this problem with acrodermatitis enteropathica. And Sheila Mefta, my daughter, she took care of a family from Abu Dhabi. And it turns out that they had two unique mutations in ZIP4, which was published later. Now, if you look at the zinc homeostasis has to be very tightly maintained. Now, this is a picture of a very severe zinc deficient boy, and he has no hair, and he had bald. He was very sick. He had a lot of infections, and once we treated him with the zinc, this is the way he'd look like at the age of 16. Now he was going to college. So it's a remarkable effect of zinc supplementation in this disease. Next slide. The other download. Well, there is another transporter, ZIP13, and ZIP13 mutation causes a disease called Ehlers-Danlos syndrome. It's a very devastating disorder. Again, the subjects do not live for a long time. Their joints are very hyper-flexible, and their connective tissue and bone metabolism are affected very adversely. This was reported by Dr. Toshi from Japan a few years ago. And we think that, although I'm not sure that by giving a lot of zinc to these patients have helped clinically, I'm not sure about that as yet. Not many trials have been done. It's also a rare disease. But it will be interesting to see if one could catch these patients very early when they are growing and make a diagnosis and give them zinc. Perhaps their bone and connective tissue disorders might be affected and might be reversed. Next slide. Okay, this Wilson's disease, you heard a little bit, Mike told you about Dr. Brewer. It so happens that it's a mutation of ATP7B, and because of this mutation, the patients accumulate a lot of copper in their body. It goes to the liver, it goes to the brain, goes to the kidneys, and indeed they never lived beyond the age of 40 to 50. They will die because of liver disorder or brain disorder, kidney. And it appeared, we were doing some studies to see if zinc will compete with copper and decrease the copper burden. And our studies for many, many years showed indeed that if you give zinc to these patients, their copper load will go down. And now, actually, there are about 200 patients that George Brewer started to follow all over the country, and with excellent results, so much so that FDA has approved copper excess treatment, in Wilson's disease to be treated by zinc. And this is now FDA-approved treatment. And it's being used all over the world now, treatment of Wilson's disease with zinc supplementation. Okay. Now, I was a hematologist, and I saw a lot of sickle cell patients in my clinic. And one day, a patient, he was 20 years of age. My resident brought him to my office, and he looked like an 8-year-old boy. I said, "Who is this?" I said, "Well, he has sickle cell disease, and he's not growing, and he has a lot of infections, and they get a lot of pain crisis." And my first impression was that he's a zinc deficient dwarf that I saw in Iran and Egypt. Just looked like that. Well, it turns out that our studies showed that because of hemolysis, they were hemolyzing the red cells because of hemoglobinopathy. The substitution was in 6th amino acid in beta chain. The red cells would be destroyed, and a lot of zinc was coming out in the urine, hyperzincuria. And I decided that perhaps we should look into the treatment of sickle cell disease with zinc. And again, hematologists were not accepting this idea, but it turned out that at this time, it turns out that zinc supplementation to these patients is the only way that you can decrease pain crisis and sickling and improve the health of the... This was reported in Cochrane's review in 2013. And it is now well accepted that zinc treatment is very valuable for sickle cell. Okay, the next one. This one is this is the Cochrane review I'm quoting here. It came out in 2013. Thank you. And this one is, okay. This is also a very interesting story, age-related macular degeneration. I was invited by Eye Institute to advise them. They had postulated that macular degeneration is due to oxidative stress in macula. And because of this oxidative stress, the macula was being affected adversely. And they were planning to treat with vitamin C, vitamin E, beta-carotene. These are the known vitamins and all that they had effect on the oxidative stress. But I told them that, you are missing a very important one, that is zinc, because all our studies have shown that zinc is a very important element that will decrease the oxidative stress. So they accepted my idea and they added zinc. And after 10 years of a study, they found that indeed those subjects who received zinc alone, they had equally important effect on macular degeneration. They had decreased blindness and the disease was not progressing rapidly. And this is very well documented by National Eye Institute at this time. But most importantly, they also reported in one of their papers that the mortality of the patients was much decreased when you gave them zinc supplementation. And I was very curious to find out why is it that there are mortalities affected. And it turns out that indeed the effect of zinc is on the cardiovascular and events. For instance, they were getting less heart attack, less thrombosis of the coronary arteries and so on, and they were living. As a matter of fact, as you may know, that a lot of physicians now recognize that the atherosclerosis is due to chronic inflammation. And this is the only thing that they are doing about it is just to give aspirin, but they have no other treatment to decrease. And I suggest that perhaps one should also consider giving zinc to all these patients so that you could decrease the mortality. Next one. Okay. Now, this is, we reported several well-controlled, placebo-controlled studies to show that if you take zinc lozenges, you got a common cold, you have at least 2 symptoms, and take lozenges within 24 hours of the onset of common cold, and give them adequate amount of zinc. And thirdly, you should give a salt that is easily releases zinc in your mouth and nasal mucosa. Then they will improve it will be almost 40 to 50% improvement in common cold. Instead of having common cold for 10 days, they may be cured by the time their 4th day or 5th day. So we reported these 2 papers in the Annals of Internal Medicine and also in Infectious Disease. And some of the controversy that remains in this area is because they do not use the proper salt. They use zinc lozenges which contains lysine, and lysine will bind it will bind zinc and will not release it to be effective in the mouth and the nasal mucosa. And also the dose was not adequate, and the time. If you don't give it early enough, it is not very effective. You must do it within 24 hours. So that's the story that we published in a couple of journals, well-controlled placebo trial. Next one. This. Oh, yeah. Okay, this one is also a very interesting area. It turns out that if you take elderly subjects in America, Michigan, and if you tested them for zinc deficiency, you will find that almost 30% of these elderly subjects are zinc deficient. And this is a very large number. And no one thinks that they should be, because they are healthy, they are not alcoholics, and they have good diet and do exercise and so on and so forth. But they were zinc deficient. It turns out in our study that their diet consists of a lot of cereal protein, which contains a lot of phytate. This organic phosphate compound binds zinc and prevents it from getting absorbed. And we think that in the elderly, is the phytate content in their diet is increased. And we have very good dietary data in these patients to show that indeed that is the case. But just think of it, the enormous significance for our elderly population. These are the ones who get very sick. They have inflammation, they have cancer, they have Parkinson's disease, and so on and so forth. And these are all due to the fact that oxidative stress and infection is increased in the elderly subjects. Next one. This one is okay. Now, as luck would have it, I got very sick myself. March 31st. I was so sick I could not get out of bed. I could not eat. It was difficult to stand up and balance. And I thought that I was suffering from some neurological problem. But I called my physician, my personal physician. He said, Dr. Prasad, I'd like you to test for COVID. I said, well, it's impossible. Why would I get COVID? I have not gone to my hospital since March 16th. But to my great surprise, I found that I was positive. It was COVID-19. Now, they wanted to send me to hospital, suggested that I should go there, but I refused to do that because I knew that if I go to hospital, I'll be completely isolated, and even if I die, nobody will come and see me. So I told my physician that no, I want to go to hospital, I will stay home, but treat myself. And the idea came to me that perhaps I should use 50 milligrams of zinc twice a day. And the reason why I selected that, because it turns out that the COVID-19 requires RNA-dependent RNA polymerase to survive, and to survive and live and so on. And turns out that zinc is a very powerful inhibitor of this enzyme. And this I had, it was reported in the literature. So I thought that, my God, this is a very good killer. Why not use zinc? And the second thing I found out that from our own studies, that zinc deficiency decreases T cytotoxic lytic cells. And if you give zinc, you can enhance T cytotoxic lytic cells, and they are powerful killers of leukemia antigen, lymphoma antigen, and also COVID. So these two things in my mind, I thought that, well, I cannot find any better killer for COVID-19 than zinc. And then I also decided to use this hydroxyquinoline, because this is a drug that has been used for malaria treatment for many, many years. But it turns out that it is a zinc ionophore. It brings zinc from outside to inside the cell. So if I give zinc and give 5-hydroxychloroquine along with zinc, I will enhance the level of zinc inside the cell, and it will be a very powerful killer. So those were my two major theses, and depending upon that, I started treating myself with hydroxychloroquine and zinc. It turns out that at that time, the pharmacy will give you only hydroxychloroquine for 5 days, and they stopped me from getting more hydroxychloroquine, but I told Dr. Gadoshian that no, I want to continue it for 2 weeks. And sure enough, on 10th day, I saw a remarkable improvement. I was able to stand up, I was able to swallow food. I was not completely normal, I didn't have all the strength, but I was 50% better at that time. And by the time 10th day was over, I had recovered almost 60-70% of my strength. And so this is the story. Now Detroit News published this and this news, and then I got call from several of my friends, and I immediately suggested that perhaps we should use this as a therapeutic model for treatment of zinc. And I wrote to NIH and all that, but several studies were done, like in Los Angeles, almost 2,000, 3,000 subjects were treated like this, and they did very well. And so also in New York, a Russian-born physician, he treated 900 patients, and they were all doing well. The problem has been that NIH has not accepted this because as a treatment, because this was not a double-blind placebo-controlled study anywhere. And they said that it has to be done. Now, I immediately wrote a double-blind study protocol and sent it to FDA, CDC, and told them that I am ready to do it in Detroit. Give me permission. And you wouldn't believe it, my application is still waiting in FDA for action and CDC. Very disgusting. I do not quite understand this, but a lot of interesting things are happening. One is economical, because the vaccine people were coming out very strong. They were spending billions of dollars in developing vaccine. And they thought that, well, if zinc is approved as a treatment, it will kill their entire industry. So one professor from Chicago, he wrote an article in Lancet, and he said that hydroxychloroquine is the worst drug that you can ever use, and it's very toxic, and so on and so forth. It turns out that all his data were false. He had just made it up in his laboratory, and as soon as it was recognized that he had false data on which he reported that finding, Lancet withdrew that article, and he lost his job at the university. So this is what happened. But nonetheless, FDA and CDC and NIH, they're still sitting on my application as of now. Hopefully, I will be permitted to do that. Next one. What is it? Oh yeah, I do that. Thank you. Biomarkers, yeah, okay. One problem with zinc study in the world has been that there is no simple biomarker for zinc deficiency. Until 1965, we were working with dithiazone technique, which was so difficult to do. And then it was most laboratories did not want to even touch it. So no one was doing any work. Now, then in 1965, we reported a technique for zinc assay by atomic absorption spectrophotometer. And this brought the technology to microgram level. Now you can measure microgram per ml. And it was simple. And actually all over the world, even now, they are using atomic absorption spectrophotometer, although it's not very popular, but it's being used by many, many laboratories. The problem is that it's still a kind of not a very good biomarker for zinc deficiency. Next one, let's see, what do I have here? Which one? Oh, yeah, okay. Now, this is something that I want to point out right here, is that in our human model, we looked at many, many things very carefully, and we found that perhaps the thymolene activity was the most sensitive biomarker of zinc deficiency. For instance, we will bring these patients, put them on a zinc deficient diet. By 8 weeks, the thymolene activity will go down. And with respect to the zinc level in the plasma, it will take 6 months before it will show a decrease. But if thymolene activity was going down, we looked at IL-2, mRNA, natural killer cells and all. They were all being affected much before you saw any change in the plasma. Also, we looked at the lymphocytes and all. So thymolene activity will go down in 8 weeks, the lymphocytes in 20 weeks, and plasma will be 24 weeks. So that tells you the sensitivity of these biomarkers. Yet, how many people can do thymolene activity? That's another issue. Next slide. We were able to do the thymolene activity in an animal model. And now we are thinking about using a new technology, which I will show you about right now. This one, what? Oh, Nelsie, yeah. My collaborator and friend, Dr. Fredrickson, Chris Fredrickson, he's here also in the audience. He developed a X-ray transmission technique to measure zinc in solid material. He will take a small piece of nail, put it under the machine, and it will immediately, within seconds, give you the level of zinc in the nails. And he utilized this technique to do the elderly zinc in our study. And he concluded that perhaps the measurement of nail zinc in the elderly is the best clinical biomarker of chronic human zinc deficiency. Now, the problem is at this point that we are interested in looking at also the zinc in plasma, in the red cells, in the lymphocytes, and all that. And that is not possible until we develop some techniques. And that's what my goal is going to be for the next few years. And once we succeed with that, this will be a nanotechnology from microgram to nano, 1,000 times more sensitive. Next slide. Okay, now very quickly I will summarize here that with this technique, we found that nail zinc is there a certain level, and if it is below that, it's deficiency. Above that, it is sufficiency. And then it turns out that it is very deep, very much, it correlates very well with the phytate zinc molar ratio, suggesting the second point that perhaps the chronic zinc deficiency is related to increased phytate in the diet. And we are pursuing all these ideas. Next one. Okay. Okay. All right. So anyway, in conclusion, I want to tell you that how the entire field of zinc metabolism in a human emerged from a single observation in one patient in Iran, Egypt. And now it has shown that there is a tremendous explosion knowledge in biochemistry, molecular biology, and immunology. And this explosion is still going on at this time. Particularly, there's no question that zinc is involved in zinc signaling process. The zinc is giving message inside the cell to do a lot of molecular events, which describes the function of the cell. And I think it's a very exciting area to pursue. Well, I can predict that we have seen a lot of excitement so far. And now I think next 5 decades, we will see another excitement in the zinc field, and that will have to be molecular signaling of zinc inside the cell to understand how the cells are functioning. I have had many collaborators mentioned. I want to thank all of them, and the two of them are here, Sheila and Chris Fredrickson. And it's really, they have been of great help to me throughout all these years. I must also thank Sally Bates, my secretary, who has been of great help to me. My restriction of vision has really impaired me quite a bit, but with her help, technology, improvement, and I'm able to look at the reviews, look at the studies, and do some work. So I want to thank Sally Bates also. Thank you very much.
Thank you so much, Dr. Prasad.
Thank you. Thank you. Thank you.
As you can tell, it's very hard to capture 70 years of amazing scientific discoveries in a 45-minute lecture, but I think you did a fantastic job. And as you can see, Dr. Prasad is still doing tremendous and cutting-edge work in the field. So I think we owe Dr. Prasad and his colleagues and Dr. Brewer a tremendous debt of gratitude for all of the work that they have done and are doing. And let's, again, just celebrate with him his induction into the Nutrition Hall of Fame.